Okay, so two questions. Why is water able to pass through this hydrophobic core if it is polar? Right? Okay. Why do you think so? Why is water able to pass through the hydrophobic core even though it's polar? Then let's look at the factors. Alright? So polarity, size, and concentration, all three plays together. Indeed, it is polar. That's why it takes so long to pass through. It's slow rate of passing through because it's polar. However, one thing is on the side of water. It is really, really small. Okay? So because water is really small, it can still slip through, but you can expect that it's going to be really slow too. Alright, because it doesn't quite like to interact with the hydrophobic core. But it's small enough. By chance, some of the water molecules may have enough energy to force its way through. Okay, we're going to watch a short video now to show you the last form of transport. This last form of transport helps us transport substances up or against the concentration gradient. But it requires one special factor. In order for us to achieve this, we require energy. All these processes that we've talked about so far, 1, 2, and 3, doesn't require energy at all. It's a very passive process. This last process, however, requires energy. It's also one of the reasons why we need to generate energy in our body. But sometimes we have to transport things against the concentration gradient. So we're going to watch a quick video for this last form of transport. Okay, sorry about the color. The color today is not quite ideal. Oh, is there sound? Okay, give me a while. I think the sound also off.
transport, active transport, or bulk transport.
May I get you to, you can ignore these three forms of transport. The way we look at it in NJ, where we break it down even further. Okay?
It's the movement of substances into or out of a cell without the expenditure of energy by the cell. One form of passive transport is diffusion. During diffusion, molecules move across a membrane from an area of high concentration to an area of low concentration. The molecules are therefore said to be moving down a concentration gradient. This continues until equilibrium is reached and the molecules are distributed equally. Another form of passive transport is facilitated diffusion. Facilitated diffusion occurs when an ion or molecule diffuses across a membrane faster than expected, either by way of a specific channel protein or with the assistance of carrier proteins that change shape as they pass through. The diffusion of water across a membrane, or osmosis, is another example of passive transport. In many cases, specialized proteins called aquaporins allow for the more rapid transport of water molecules. The second type of transport, active transport, requires the input of energy in the form of ATP. The proteins that conduct this form of transport are often called pumps because they force molecules, or ions, to move from an area of low concentration to an area of high concentration. This is commonly referred to as up or against the concentration gradient. One of the more common examples is the sodium-potassium pump, which moves sodium ions back out of the cell and potassium ions into the cell. Notice that the sodium-potassium pump undergoes a change in shape that allows it to combine alternately with sodium ions and potassium ions. The third type of transport, bulk transport, is used for molecules that are
Okay, so that was the last form of transport, active transport. Okay, so the word is active transport. Can you find it in your notes? Okay, because it's not in your class activity worksheet. Can you search your notes for this particular part on active transport? Okay, just so you know where you can find it up. Okay, so it's on page 18 of your notes. This way you can find active transport. Okay, I do not need you to go and memorize and make notes for this particular section. Alright, I don't need you to define all these forms of transport. It is more of a skill. Instead, I will very likely show you a random biomolecule. Then I'll ask you, which form of transport do you think will need to take place? Okay? So don't go and waste time rewriting all of this. Okay, it is a skill rather than content here. Okay? In the lab last week, we spent some time playing potatoes. Did we? Yeah? Okay. Okay, we play potatoes. And also you play with the cutouts, right? Where you look at vesicles. Vesicles is another way of transporting substances across the membrane. You have seen it last week. It helps us transport really big things.
Too large to be moved by transport proteins. Instead, vessels take them into or out of the cell. During this process, the plasma membrane surrounds and engulfs the particle.
So if we have really big things that are not even as big as glucose, are way bigger, like proteins, well, we have to use a much drastic process. And that process we kind of played with last week in the lab when you're trying to get vesicles to fuse together. Right? So there are two ways in which substances can enter and leave the cell if they are too big. This process is called endocytosis, if you want it to go in. Endo means inside.
This is known as endocytosis. Cells use three basic types of endocytosis.
Okay, and if you want to get things out,
fuse with the membrane.
it will be called exocytosis.
Outside the cell.
Where can you find that in your notes? It's on the next page. If you scroll down a little bit more, you'll see vesicular transport, endocytosis and exocytosis, also on page 18. This form of transport, we activate it if we want to transport really big things. Or if we want to transport a lot of things at one go. Okay, so we just covered quite a few methods of transport. This is also more of an application kind of chapter. Don't expect us to just test you what you see written in words there. It's skills based. We'll provide you a context and then we'll ask you which method you think we should use. Okay, with that, we've come to the end of this particular chapter. There is a TE on transport, TE 3.1. Okay, may I get you to do it by this weekend? Yeah, TE 3.1. Yes. Oh, like this one's over here? Could it be broken down? And they could be. So actually, one way our cell membrane is able to regulate how many things can go in and out is whether how many of these proteins are present on the membrane. Let's say I want more substances to be transported in and out. I could embed more of these glucose proteins, transport proteins. If I want less, then I'll just remove them away. So that's one smart way our cell membrane is able to regulate the amount of substances going in and out. Okay, can I give you some time now to consolidate your notes? If you have anything new you took down, can I get you to write it down inside your notes? I think I may have missed out on one. Yes. These fellas? Right. Do you know what the cell membrane is made up of? Yes. Okay. It's actually from your chapter on lipids. Yeah, okay. Okay. But it's a specific kind of lipid. You go and find out there. Okay, you scroll back to your CA or you go back to your notes on lipids. You go and find out what this lipid is. Okay, can I get everyone to go to page 18 of the notes? Okay, sorry, this is not 18. Page, whatever this page is. 14. Go to page 14 of your notes. You see these two bolded words? Carrier proteins and channel proteins. Can I just get you to highlight that? Okay, so far, I've been referring to these fellas as transport proteins. I'd just like to introduce you two kinds of transport proteins that you may encounter across biology from this day on forward. You can encounter two kinds. One is called a channel protein, and another is called a carrier protein. I'm going to draw them slightly differently. So a channel protein will often look like this, with a paw. Okay, so this is a channel. A carrier protein often looks like this when drawn in a diagram. I'm going to show you a short video just to let you see the difference in the channel and carrier protein. They function differently, but both serve the same function to help things move across. Okay, this is what a carrier this is what the two proteins
There are two main types of transport proteins, channel proteins and carrier proteins. Channel proteins are tube-like and provide openings through which small, dissolved particles, especially ions, can diffuse. In general, each type of carrier protein has a shape that fits a specific molecule or ion. When the proper molecule binds with the protein, it changes shape and moves the molecule across the membrane.
Okay, so that's it. A carrier protein can change shape. Okay, it's very cute. You can change shape to allow substances to pass through. Channel proteins are just like straws. Things can just slip through without needing to change any shape. So if I just ask you, an aquaporin, which kind of transport protein do you think it falls under? Aquaporins. Sorry? Okay, it's more like a channel. Aquaporins are more like channels. Okay? They are just like a tube, and then water molecules can pass through. Yes? How? Okay. Usually, unless we tell you exactly what kind of membrane protein it is, then I would say you don't have to. You can just tell us simple diffusion of the stated diffusion and so on and so forth. Yeah? Unless you actually know. Sometimes you will know whether it is a carrier or a channel. As the biology chapters unfold, we will sometimes tell you exactly what kind of protein it is. Right? So for example, there's one that you will confirm need to know by now. It's for water. Water requires a channel protein called aquaporin. As we move on, you will slowly see other kinds of proteins. Okay? Yes?
How are you supposed to know if it's a channel protein or...
Okay, good question. how are you supposed to know if it's a channel or carrier protein? You look at the shape. So usually channel proteins are drawn in a certain way. I'll show you another video, perhaps it will help you see how we can otherwise depict it.
How do materials enter?
Okay, so you look at this particular diagram over here. Okay, can you see? Channel proteins often, they have a straight channel through it. No blockage usually. So if I let this video run out, you can see that things can just pass through without obstruction. Channel proteins. Yeah. Carrier proteins often look like they have like a carrier proteins often look like they have like almost like an enzyme like that. Okay, often looks like a clip. Okay, that's what carrier proteins often look like. They look like a clip, and this clip can change shape to allow substances to pass through. Okay, thank you. I think that was an important point to take note of. What do these proteins actually look like? Okay, we have around 15 more minutes before the lesson ends. Okay, I don't think I will be able to do the start next chapter meaningfully. May I get you now to begin on your TE for transport across membranes? Okay, start on it or do as much as you can. Yes. Yeah, what? Oh yeah, okay, I'll come over in a while. Okay, so can I get you to begin on your TE as much as you can? Okay, there are quite a number of MCQs. How about you complete at least the MCQs for today? And if you're up for it and you're fast, might as well complete the structure today also. Okay, but MCQ within class time, can I get you to complete it? Yeah, sorry, how? Okay, or maybe I put it this way. Active transport typically makes use of carrier proteins. Okay, but it is not correct to say that carrier proteins are always used for can be used for facilitator. Right, then I will. Oh yeah. Okay, wait, it's downstairs. Yeah, I come out and I'm downstairs.
Mr. Tan,
Yeah.
they are both, the channel and carrier proteins are both for facilitated diffusion.
Oh, sorry, what?
Both of the proteins are facilitated diffusion.
It can be used for facilitated. Okay, then what about active transport? So active transport, right, also can make use of these proteins. Oh, what's the point? That's a good question. I do feel that when you look at the carrier proteins, right, there's a little bit more regulation. Yeah, because the correct thing needs to bind to the thing first, the transport protein first, then you will change it. So I feel like there's a little bit more fine regulation. If you don't know the meaning of hyper and hypotonic, can I get you to Google it? It is related to salt concentrations and water potentials. So just a quick Google of what hyper and hypotonic means.
네.